Hey everyone, welcome back. In today's video, we're going to talk about FASTA files. If you're doing genomics or bioinformatics work, you'll definitely be working with these files. Let's get started. So what is a FASTA file? It's a file format for storing well-characterized biological sequences. So things like the DNA sequence for each human chromosome, RNA sequences for things like protein coding transcripts, or extra chromosomal sequences such as plasmids. And you'll know when you see a FASTA file because they typically end with a .fasta, .fa, or .fna file extension. So where do FASTA files come from? They actually come from the reads in a FASTQ file. And what you can do with these reads is assemble them into longer sequences. It's sort of like taking pieces from a puzzle and trying to fit them together until the puzzle is completed. And information about the assembled pieces and the entire puzzle is what's stored in FASTA format. And this whole process is referred to as genome assembly. And there's an entire field of bioinformatics dedicated to just this topic. And I'll make sure to provide some links to useful papers in the description about genome assembly for those of you interested in learning more about it. And the FASTA format is actually quite simple. Each entry within a FASTA file consists of 2 lines. The first, a descriptive name of the genetic feature. Again, this could be the name of a chromosome, a gene, or bacteria, which always begins with a greater than sign. The second line is the biological sequence associated with the genetic feature. And here's an example of a real FASTA file. This is a FASTA file for the Staphylococcus aureus bacterial genome and its associated plasmids. You can see how each sequence is labeled with a descriptive header, beginning with a greater than sign, and followed by the biological sequence itself. So what are FASTA files used for? So they're used for storing high-quality reference data sets. For example, human and mouse genomes and their associated genes. Cataloging genomic diversity across the tree of life. For example, reference genomes for plants, animals, bacteria, and viruses. And most importantly, they provide us something to compare our data to, which is really useful for things like identifying sequence variation, so things like single base substitutions, insertions, and deletions, counting microbes in a microbiome experiment, and quantifying gene expression in bulk or single-cell RNA experiments. You need a reference genome for all of these things. So these are very, very important files and something you'll be working with a lot. So where can we download FASTA files? There are numerous online resources where you can find these files. GenCode, which stores human and mouse reference genomes. RefSeq, which has over 165,000 reference genomes for bacteria, fungi, plants, animals, and viruses. GIS-AID has over 21 million viral reference sequences. And CARD has over 6,000 reference sequences for antimicrobial resistance genes. Now let's look at a real-world example involving the International Space Station and the microbiome. This is a cool study where astronauts collected samples from their environment while aboard the International Space Station. These samples were then brought back to Earth, where they were sequenced, and the reads compared to the RefSeq reference database, which contains genomic information about all known bacteria and viruses. And using this reference database, the researchers found that over 800 species of bacteria were present inside the International Space Station. And if you'd like to learn more about this study and where these microorganisms came from, I'll post a link to the paper in the description. That's it for this video. In the next video, we'll be talking about another important file type called SAM and BAM files, which are used for storing sequence alignment information. You won't want to miss it. I'll see you there.